In this video, we're now going to talk about the components of translation. So what molecules are necessary for translation? And the first is we need, of course, the messenger RNA. That's going to be our template. That's what's going to be read. Then we need charged transfer RNAs or tRNAs. What it means to be charged is that they have an amino acid covalently bound to them. And finally, we need the ribosomes. Those are going to be coordinating this entire process. Now, there's lots of other initiation factors and elongation factors that we're going to discuss in the next lecture, but these are the three main things that we need at the start of translation. So let's take a look at messenger RNAs, and let's look first at prokaryotic messenger RNAs. And remember that many prokaryotic messenger RNAs are polycistronic. So for example, if we look at the trip operon or the lac operon, even though it encodes multiple genes, it's still one long messenger RNA with several start and stop codons. Start and stop, start and stop. So the question is, how does the ribosome know that even though this isn't the first AUG, that it's still a start codon? Because remember, in eukaryotic translation that we'll go to in a minute, we'll look at it in a minute, the ribosome binds at the 5' end, scan, that first AUG, that's it, done, start. There is no other start codons, they're just normal AUGs that encode methionine. Prokaryotic messenger RNA is different. It's got these start codons that are in the middle of the messenger RNA. How does a ribosome know that? Right before each start codon, there is something called an RBS, a ribosome binding site, a Shine-Dalgarno site. These all mean the same thing. And the sequence here in these RBSs, in these ribosome binding sites, is reverse complementary to one of the ribosomal RNAs, specifically to the 16S ribosomal RNA. So that the ribosomal RNA actually binds to an RBS, and when it does so, it knows that that very next AUG is a proper start codon. So unlike eukaryotic translation, in prokaryotic translation, ribosomes don't necessarily start at the 5' end of the messenger RNA. They can jump right in the middle of a messenger RNA when they are attracted to one of these RBSs and then start from there until they hit the very first stop codon. Another ribosome might jump right on here, hit the start, and then end at the stop codon here. So polycistronic means it's one mRNA with multiple open reading frames. An RBS is a ribosome binding site that attracts ribosomes to internal start sites. Now, eukaryotic messenger RNA is very different. It's not polycistronic. One messenger RNA is going to encode one protein. The start codon is always going to be the first AUG starting from the 5' end. Now, it can be in any of the three reading codons, or the reading frames. It's going to be dictated by where that first AUG is started from the 5' end. Don't forget, though, that a eukaryotic messenger RNA is also going to have a 5' cap, and it's going to have a poly A tail. That'll become important a little bit later. Now, transfer RNAs are about 73 to 93 nucleotides long. Like all RNAs, they are transcribed from a gene within the DNA, and then many of them are post-transcriptionally modified. That's where you see these strange looking bases here and here and here, for example. Now, it folds into this clover leaf-like structure. The only two parts I want you to be aware of is that the 3' end of it, that's where we're going to have a covalent bond between the tRNA and the amino acid that it is bound to. And the anticodon arm right here, we have our 3', and then we have our wobble position right here in the 5' most position of that anticodon. Now, there are only 32 transfer RNAs within eukaryotes, yet there are 64 codons. So why are there not 64 transfer RNAs? I'll give you a few seconds to think about that. Did you figure out because of the wobble effect? Did you figure out the fact that some tRNAs only can bind to multiple codons? The fact that if that 5' most position is in inosine, that it'll bind to 3 different codons? All of those reasons are why we only need 32 tRNAs and not 64. Now here's a more realistic look at the structure of a tRNA. A tRNA that has an anticodon that binds to a codon that encodes phenylalanine will be referred to as a tRNA phenylalanine. Now, if that transfer RNA is actually bound to a phenylalanine amino acid, like it is here, then we say we put this phenylalanine in front of it. So phenylalanine, tRNA, phenylalanine means it is a tRNA with an anticodon that binds to a codon that encodes phenylalanine and is indeed bound to a phenylalanine. It's important to know this sort of nomenclature because some of the questions that you might get might kind of mix this up a little bit. Now, the enzyme that catalyzes the charging of a tRNA, meaning the enzyme that catalyzes the formation of a covalent bond between the amino acid and the 3' end of a transfer RNA, is called an aminoacyl-tRNA synthetase. And each aminoacyl-tRNA synthetase binds to a single amino acid. So this one here, this aminoacyl-tRNA synthetase, is an alanine aminoacyl-tRNA synthetase. It only binds alanine. So there's 20 different synthetases, one for each amino acid. Now here's the important thing. Some of these synthetases can bind more than one transfer RNA. And when we're talking about different transfer RNAs, we're also talking about different anticodons. And no matter which tRNA binds to this synthetase, it is going to be charged with alanine, which means if we have multiple tRNAs binding this synthetase, all of which are now carrying alanine, all of which have different anticodons, that means that any codon that those different anticodons can bind to encode alanine. And there, folks, is your third of 3 mechanisms necessary to allow for redundancy in the genetic code. The first one was the wobble effect, the second one was inosine, and now the third one is called isoacceptor tRNAs. That means transfer RNAs that are all the same, the substrates for the same synthetase, and therefore carry the same amino acid. Let's take a look at the reaction necessary for the charging of a tRNA. What happens is this. Here's our aminoacyl-tRNA synthetase, and it has an active site for the amino acid, remember, a single amino acid. It then has a site for ATP because that ATP is necessary because we're going to activate the amino acid by adding an AMP to it. Now that activated amino acid is going to be added to a tRNA, and remember, many of these synthetases have multiple different tRNAs that it can bind to. The only commonality between these tRNAs is that their anticodons, as different as they may be, all bind to codons that encode the same amino acid, and that's why they are being acted upon by the same synthetase. Now, once the reaction occurs in which that amino acid is added to that transfer RNA, it is released and it is said to be a charged tRNA. Let's take a closer look at the reaction that occurs here to charge a transfer RNA. First, we have to look at the activation of the amino acid, right? Otherwise, the next reaction is going to be endergonic. So what we have here is the alpha carboxyl group of the amino acid is going to be our nucleophile that attacks the alpha phosphate group of ATP. And our byproduct is our pyrophosphate there, and our product is our adenylated amino acid. Next step, our adenylated amino acid is very reactive, so in comes our tRNA. The 3' end here, so that 3' hydroxyl group is going to attack again that same carbon that now has this fantastic leaving group. That fantastic leaving group leaves, AMP, and now you get a covalent bond between this O and this carbon of the alpha carboxyl group. And so here we have an extremely reactive bond. Right? Essentially, we've got an amino acid with a massive transfer RNA as a fantastic leaving group. Now, there is talk, sometimes you might hear the idea of the second genetic code. And what that means is that in order for the genetic code to work, the various aminoacyl-tRNAs have to make sure that they can distinguish between the 20 different amino acids and bind the correct one, and distinguish between about 32 different tRNAs and determine which ones of those, right, because it can be multiple of them, it is supposed to bind to. That's really important, because if you make a mistake, if you add the wrong amino acid to the wrong transfer RNA, you're going to keep building incorrect proteins. So the way in which a synthetase recognizes transfer RNAs is sometimes considered the second genetic code. Now, one might think that the majority, the real way, or the only way that it differentiates one transfer RNA from another is at the anticodon. And you'll see here these yellow circles. The bigger they are, the more likely they are being used by tRNA synthetases to differentiate one from another. But you can see there's all sorts of other areas on a tRNA that allow that synthetases use to differentiate between the 32 different tRNAs. Now, it is absolutely essential that these aminoacyl-tRNA synthetases are accurate. And there's 2 successive steps that make sure that it is indeed accurate. The first we could think of as 2 sieves. The first sieve is the active site of the synthetase and what amino acids can fit into that active site. Well, most of them can't, but there's several that can. So that's the job of the second sieve, the editing site. So now all of them that do fit, they're all going to get charged, but the ones that were improperly charged are going to fit into this editing site and then be hydrolyzed and tossed out. So let's take a closer look at these 2 sieves. Here we have isoleucine-tRNA synthetase. It's only supposed to charge tRNAs that recognize or that have an anticodon that binds to a codon that encodes isoleucine. So what we see here is isoleucine fits, that's great, but so do smaller amino acids like valine. Okay, the first thing is that isoleucine fits 200 times better than valine, so there's already a pretty good sieve. And then all the amino acids bigger than isoleucine, they can't even get in there. All right, next, isoleucine gets in there, valine unfortunately also gets in there. Both of them become charged. So ATP is going to be wasted here. Okay, valine gets charged, as does isoleucine, but that's supposed to happen. Then there's this editing site that is too small for isoleucine, but big enough for everything smaller than isoleucine, like valine here. So valine now that it's charged, it fits into this editing site, it hydrolyzes the valine off of the transfer RNA and ejects them. So the only thing that survives the charging, the addition of the amino acid to the transfer RNA, is isoleucine. Okay, so that's the 2 ways in which synthetases are accurate. All right, the next component is ribosomes. Ribosomes account for about 25% of the dry weight of a cell. So it's pretty prevalent little molecule, or big molecule, big complex. Oops. Now, whether we're talking about bacterial ribosomes or eukaryotic ribosomes, they're both made up of a large subunit and a small subunit that only come together when they're bound to messenger RNA. And both of them are composed of both proteins and RNA. Proteins and RNA. But take a look at this. This large subunit is only composed of a 5S ribosomal RNA and a 23S ribosomal RNA. So only 2 RNAs, but yet 36 proteins. Same thing here. We have 1, 2, 3 RNAs and 47 proteins in this large subunit. Small subunits, the same kind of things going on here. 1 RNA and then 21 proteins. 1 RNA and then 33 proteins. And yet, it's about 2/3 of the mass of a ribosome comes not by the proteins, but by the RNAs. They're that much bigger than the proteins. And also, the actual enzymatic activity, the formation of a peptide bond between adjacent amino acids is catalyzed not by some of these proteins here, but in bacterial ribosomes by the 23S ribosomal RNA, has peptidyltransferase activity. The 28S RNA, a ribosomal RNA, in the eukaryotic ribosome has peptidyltransferase activity. Okay, so it's the RNAs that are the really important thing. The proteins simply stabilize the structure of the RNAs. Now that peptidyltransferase reaction looks like this, and I want you to know what the electrophile is. I want you to know what the nucleophile is. Here is our charged, amino acid, or charged, tRNA. It's got the amino acid right here. Here's the transfer RNA. Now the nucleophile is the alpha amino group that is going to attack the alpha carboxyl group of the end of the polypeptide chain that is still connected to the previous tRNA, which means that we're going to have that bond broken, that transfer RNA goes bye-bye, and now we have a covalent bond between this nitrogen and this carbon. That's our peptide bond, in fact. But notice, this incoming amino acid is still bound to its tRNA. It's still bound to its tRNA. Okay? And here is that peptide bond right there. So if we take a look at an E. coli, so a bacterial ribosome, when it is in the presence of a messenger RNA, we see that the large subunit comes together with the small subunit. We have these 3 docking sites, the aminoacyl site, the peptidyl site, and the E site, exit, so the E, P, and A sites. And then through that is strung the messenger RNA. And what these docking sites are for are the incoming transfer RNAs that have amino acids attached to them come into the A site, and their anticodons will bind to whatever codon is right here, okay, if there's enough, if it is reverse complementary. And then the whole ribosome can shift down to the next codon, and the whole process will continue over and over again. And we're going to take a look at that mechanism in the next lecture. Okay, so again, next lecture, mechanisms of translation. How does the ribosome and how do the various initiation factors work together to start translation and to continue and end translation?